So let me introduce our next speaker, Dr. Adam Brickman from Columbia University. The title of his talk is White Matter Hyperintensities are a core feature of Alzheimer's disease, evidence from late onset and genetic forms. Welcome.
I'm really delighted to be here. My name's Adam. I'm from Columbia University, and I think I'm the only speaker at the symposium who doesn't look under the microscope. I'm a neuropsychologist, and I use brain imaging techniques in human cohorts. I've learned a ton here, and I'm going to talk to you a little bit about a sort of higher level on the neuroimaging side, evidence of white matter abnormalities that we see in late onset forms of Alzheimer's disease and in genetic forms of Alzheimer's disease. So what I'll talk about today is sort of how we think about Alzheimer's disease classification clinically or in a clinical research context and some of the caveats of that. We'll talk about vascular brain imaging in dementia and Alzheimer's disease, and then ask the question whether vascular brain injury is in fact a core feature of Alzheimer's disease. So it wasn't until the 1980s that the field sort of conceptualized Alzheimer's disease diagnostically in a uniform way. And you might recognize these diagnostic criteria from McCann et al. That listed probabilistic diagnoses based on ruling out certain symptoms. So we called our patients probable Alzheimer's disease when they had a dementia syndrome, and we could rule out any known other potential causes of dementia. We called them possible Alzheimer's disease when they had a dementia syndrome, but there was some question about the onset and course, and maybe in the presence of the context of other causes of dementia that are not considered primary. And of course, we only diagnosed definite Alzheimer's disease when we had a clinical diagnosis of probable AD, so they had to have the dementia syndrome, but they also had to have histopathological confirmation of those plaques and tangles, either at autopsy or in the rare case when someone came in for a brain biopsy. So if you fast forward, to 2010 in this influential paper by Cliff Jack, a huge thing that's changed in the field since I've been in it is that we no longer have to use these probabilistic diagnoses. We now have biomarkers where we can operationalize some of the pathologies that we expect to see on autopsy. So Cliff came up with this model, and I'm sure everyone on the call has seen some version of this before, where it stages the pathophysiological processes that happen in human Alzheimer's disease. And they start with beta amyloid deposition, followed by tau-mediated neuronal injury and dysfunction, followed by brain structure or trophic neurodegenerative changes, followed by the clinical syndrome and the clinical dementia presentation. So what's important in these models is that it's important to note that the beta amyloid abnormalities happen years before the symptom onset. And what's changed in our lifetimes has been that now we can measure some of these different pathophysiologies. So we can use PET scans and CSF and increasingly plasma and serum samples to measure amyloid, tau. We, of course, can use MRI to operationalize atrophy and use the neuropsychological examination to look at cognitive function in the clinical exam to define dementia. And these hypothetical curves were translated in 2011 into new diagnostic criteria that included preclinical Alzheimer's disease, that was biomarker evidence of AD without symptomatology, mild cognitive impairment due to Alzheimer's disease when there is evidence of abnormal biomarkers with some clinical or cognitive dysfunction, and of course, dementia or due to Alzheimer's disease when we had biomarker evidence of AD along with the clinical syndrome. And fast forward to 2018, those diagnostic criteria were revised yet again to this new research framework. And the way the new ATN biomarker framework operates is that if you have a patient or a participant or a subject, you determine whether or not they have biomarker evidence of Alzheimer's disease, of A-beta. And if they don't, then we say that they don't have Alzheimer's disease. And if we do, then we look at whether or not they have biomarker evidence of tau pathology. And if they don't, we say that they're on the Alzheimer's spectrum, or they have some evidence of Alzheimer's pathologic change. And if they do have tau pathology or evidence of tau pathology, then we say that they have Alzheimer's disease. So this is a very strict ATN model where the diagnosis of Alzheimer's disease is now a rule-in diagnosis, and it's based on biology of evidence of beta amyloid of tau without consideration of cognitive or clinical symptoms. And that's a huge change. Of course, with these new criteria, once we call someone as having Alzheimer's disease, we can classify them as being unimpaired, having mild cognitive impairment, or having dementia. So where I kind of came into the picture is I asked the question of, as many others have, does pure Alzheimer's disease actually exist in the wild? And we can turn to, we heard a little bit about the Ross-Map study, and this is work of Julie Schneider and colleagues that characterized the pathological breakdown of participants who were followed clinically for many years in their cohorts and diagnosed at the end of life as having no cognitive impairment, mild cognitive impairment, and probable Alzheimer's disease, based purely on their clinical presentation. And the pie charts that you're looking at represent the main pathological categories that they see at autopsy. And if you look at the brown pie wedge, which is the Alzheimer's disease, pure Alzheimer's disease plaques and tangles, you see that that wedge gets smaller as people shift from no cognitive impairment to mild cognitive impairment to probable Alzheimer's disease, such that and then what you see much more is a mixed profile of Alzheimer's, a mixed pathological profile where typically Alzheimer's pathology coexists with vascular pathology, other forms of neurodegenerative pathology, or is represented purely by vascular pathology. And so what we can say from these experiments or these observations is that pure Alzheimer's disease indeed exists, but typically really only in asymptomatic people. And when we look at what our patients, our participants who actually have the Alzheimer's disease syndrome, it's really defined by a mixed pathological feature. So what I often say is that the human Alzheimer's disease model is really a terrible model for mouse Alzheimer's disease, which is a much more quote unquote pure form of AD. Of course, we know from the epidemiological literature that there are a number of vascular risk factors like diabetes, insulin resistance, high blood pressure, heart disease, high cholesterol, even ApoE as a vascular gene, seem to elevate the risk, at least for clinical Alzheimer's disease. And presumably, the increased risk for AD is due to proximal vascular damage in the brain. And we think that this cumulative vascular burden may put the brain's white matter at particular risk of injury. So why would the white matter be particularly vulnerable? We know that the vascular supply throughout the brain, of course, is not uniform, and the white matter is perfused by the most delicate or the smallest vessels that are probably most vulnerable to damage or pathology. There's a ton of evidence, we've heard some over the last couple of days, that the white matter damage or damage in the Alzheimer's brain may be secondary to blood-brain barrier breakdown at the level of the endothelium. We heard a little bit from Liz Head yesterday that we see these increases in tortuosity in the vessels as they enter the white matter from the gray matter, which might also put them at particular risk of injury. So in my lab, we capture this vascular brain injury to the white matter by looking at these specialized T2 weighted MRI scans, particularly fluid attenuated inversion recovery scans. And what you can see on these scans are these blotches of white hyperintense signal, which a normal, healthy young person doesn't have and seems to accumulate with age. And we can quantitate these with software that we've developed and look regionally, et cetera. And the pathology underlying this white matter disease isn't fully well described, and it's probably quite heterogeneous, but most consider white matter hyperintensities to reflect rarefaction of the white matter secondary to small vessel occlusive disease or hypoperfusion injury, certainly something that we think is pointing to something vascular in nature. And I think that one thing that's going to be important is for folks like me to start talking and interacting more with some of the speakers that we heard to really try to unpack what we're seeing under the microscope when we look at these lesions on MRI scan. So my lab's been concerned with the questions like, are these hyperintensities as a marker of small vessel disease, a core feature of Alzheimer's disease and involved with pathogenesis or simply the clinical presentation of AD? Do they interact with other biological markers of AD and what drives those interactions? And can we leverage neuroimaging to inform our conceptualization of AD and identify potential treatment targets? So a lot of the work that I do is community-based. We use epidemiological techniques to sample older adults and middle-aged adults in the community around Columbia University Medical Center. It's really diverse in everything you can measure. There's a huge range, and it really represents the population out there. Of course, we do a lot of imaging in our cohort studies. We quantitate white matter hyperintensities. We look at atrophic or neurodegenerative changes along with a million other MRI sequences. And some of our early observations, what we found was that people with amnestic forms of mild cognitive impairment, those are the folks who are at greatest risk for developing clinical Alzheimer's disease, seem to have the greatest burden of small vessel disease operationalized as white matter hyperintensities. So this was a surprise. We thought that amnestic syndromes shouldn't go along with cerebrovascular disease. In fact, we thought that non-amnestic syndromes would be more explained by small vessel disease. When we look regionally, we can quantitate white matter disease in the frontal, temporal, parietal, and occipital lobes. We see this interesting regional dissociation. People with cognitive impairment do indeed seem to have anterior elevation in white matter hyperintensities, whether they have non-amnestic MCI, amnestic MCI, or clinical AD, but it really seems to be a posterior story, particularly parietal lobe elevation of white matter hyperintensities in people who are at greatest risk for developing Alzheimer's disease and those with prevalent clinical Alzheimer's disease. And of course, these data are very similar to what other groups have reported, like Charlie DeCarli's group in California. We can ask the question whether baseline white matter hyperintensities predict who's going to get Alzheimer's disease in the future. And we consistently see that elevation of parietal lobe white matter hyperintensities elevates the risk for future or incident Alzheimer's disease. And this is sometimes even more predictive than atrophic changes or neurodegenerative changes like hippocampal volume. And this is just a survival curve. So we've been asking the question, like, how do we get at the question of whether in human data, whether vascular disease or small vessel disease sort of hangs together with AD risk? And one way to look at it is very simply to ask normal, healthy adults if they have a family history or parental history of Alzheimer's disease. And it turns out that even in this crude analysis that people matched on every other aspect and asymptomatic themselves, people who report having a family or parental history of Alzheimer's disease, in fact, have elevated evidence of white matter hyperintensities in their brain. If we take the same folks and look at people with, again, cognitively normal, healthy older adults, and just divide them into ApoE4 positive or negative, we find that E4 carriers also have elevated white matter hyperintensities, and this effect is greatest in the parietal lobe. So irrespective of any symptoms, of any known pathology, those who are at greatest risk by virtue of having an E4 allele of developing Alzheimer's disease have evidence of increased cerebrovascular disease, particularly in posterior areas. We can use genetic forms of AD to inform the questions, and you might be familiar with the seminal study, the DIAN study, or the Dominantly Inherited Alzheimer Network. And this is a community, collaboration among a number of centers that see a lot of presenilin 1 and 2 mutation carriers. And you can use the information, and as you know, presenilin mutation carriers it's a fully penetrant mutation, and it's autosomal dominant. So you can recruit people into the study that have a parental history of presenilin 1 or 2 mutation, meaning that they themselves have a 50% chance of inheriting the disease or the gene, and a 100% chance of manifesting the disease if they've inherited it, and compare them to their siblings who have a 50% chance of not inheriting the gene mutation and therefore not developing Alzheimer's disease. And you can construct these quasi-longitudinal tables. So because presenilin 1, we know a lot about it, your age of onset, of symptom onset, is going to be about what your parents' age of symptom onset is. So I'm 47 years old. If I happen to have gotten a presenilin 1 mutation from my mom who manifested symptoms at age 50, then we would say that I'm 3 years away of developing symptoms myself based on my mutation. And so we can use these data to order people based on their estimated year to symptom onset. And in this paper from Randy Bateman and colleagues, They showed data that are very consistent with the amyloid hypothesis. Basically, amyloid deposition happens years before, maybe even 20 years before expected symptom onset. That's followed by increases in tau biomarkers, and then it's followed by increases or decreases in memory functioning. So we took data from the DIAN study and we quantitated those white matter hyperintensities, and this is work that Sanju Lee led. And what Sanju found was that in these young onset, early onset participants who were generally free of vascular risk factors, they evidenced increased, they showed increased evidence of small vessel supervascular disease in the form of white matter hyperintensities about 10 years prior to expected symptom onset. And when we look regionally, again, this is a posterior story where we see these increases in white matter hyperintensities up to 20 years prior to expected symptom onset in these mutation carriers relative to their sibling controls. We replicated these findings just recently in another genetic cohort study. This was in collaboration with Yatil Kuris's lab and led by her postdoc, Dorothy Schumacher, where we showed that in her kindred in Colombia, the country Colombia, not the university, they had elevated the mutation carriers had elevated white matter hyperintensities. This was greatest in the parietal lobes again. And these white matter hyperintensities elevated, at least cross-sectionally with age, in a manner that seemed to map or be quite similar to tau markers of tau pathology. We took it a step further and we showed that, in fact, in this cohort study, the degree of the amount of white matter hyperintensity or the white matter hyperintensity volume accounted for more variance in their cognitive symptoms than any other of the biomarkers that we measure, including measures of CSF phosphorylated tau, amyloid PET, SUVR, cortical thickness. It was really these white matter hyperintensities that seem to be accounting for the cognitive symptoms that we see in our patients. We've been working, you've heard a little bit about this so far, very closely in the ABCDS, the Alzheimer's Biomarker Consortium in Down syndrome, as you heard from Dr. Head yesterday. Studying folks with Down syndrome is very, very interesting from our perspective. It's super interesting because they seem to be protected from a number of the vascular risk factors that we think might be confounding the question that we have about whether cerebrovascular disease might be a core feature of Alzheimer's disease. So it appears that folks with Down syndrome are protected against developing things like hypertension, and they have very healthy vessels, it seems. So we asked the question, and this is work led by Patrick Lau, who's then a postdoc and now a faculty member at Columbia, and we quantitated all sorts of forms of vascular disease on MRI scans, including white matter hyperintensities, enlarged perivascular spaces, even frank infarcts, and microbleeds, which we think in this cohort probably reflect cerebral amyloid angiopathy. And what Patrick showed, again, was that there's this sort of dose response across diagnostic groups in total white matter hyperintensity volume, such that people with AD or clinical AD had reliably higher amounts of white matter hyperintensities compared with their possible AD and their mild cognitive impairment colleagues. And again, this was a regionality story where we see the effects much greater in posterior regions, including parietal, and in this case, occipital lobe. We also saw a similar effect in the other vascular markers. We saw frank infarcts, which hadn't been reported before, in up to 20% of our symptomatic participants. We see enlarged perivascular spaces early on. As soon as they become symptomatic, they seem to have enlarged perivascular spaces. And as we heard from Liz yesterday, about 40% of symptomatic Down syndrome folks have evidence of cerebral microbleeds, which is a nice parallel to what she showed in the pathological tissue, where they see about 40% of participants have identifiable microbleeds on pathology. We did some proteomic analyses. This work led by Famita Modi, a student in my lab and in collaboration with Sid O'Brien's group, where we examined proteomic markers in the plasma and looked at use support vector machines to see how they associate with these different markers of cerebrovascular disease in Down syndrome. And we found essentially that there were greater inflammatory and vascular correlates like IL-10, the adhesion molecules, CRP, TNF-alpha, that seem to correlate with some of these cerebrovascular lesions on MRI scan in asymptomatic participants. And as they moved into becoming symptomatic, we saw that the correlates were more strongly associated, that the vascular lesions were more strongly associated with markers of neurodegeneration in the plasma, like A beta 40 and 42, total tau, neurofilament light chain, and phosphorylated tau protein. We've been collaborating with Liz's lab, and we're starting to look at the postmortem correlates. And I think Liz showed something like this, similar to this yesterday, where we certainly are starting to see increases in amyloid angiopathy and in participants with Down syndrome. We're starting to look at the astrocyte phenotypes. We're looking at pericytes, and we're very, very interested in blood-brain barrier breakdown. So I think one question that we've had is whether these markers of vessel disease and A beta pathology interacted, in work led by Frank Provenzano several years ago. Frank took ADNI data, quantitative white matter hyperintensities, looked at amyloid PET scans, and what he showed was that among those with amyloidosis, or those with amyloid positive PET scans, those with clinical symptoms of AD had reliably higher white matter hyperintensity burden. And if you wanted to use white matter hyperintensities as some sort of a diagnostic marker for people who might be symptomatic, we could come up with a cut score that yielded an 83% sensitivity and a 64% specificity. He went on to show that both increased white matter hyperintensities and amyloid status at baseline predicted future risk for the development of clinical Alzheimer's disease in patients with AD and in patients with mild cognitive impairment. And this seemed to be synergistic. But we don't think that it's really a story of the intersection of vascular disease and beta amyloid. Apart from cerebral amyloid angiopathy, it seems like there's an emerging story that relates cerebrovascular disease driving tau pathology. And in work led by Batul Rizvi, former student in my lab, Batul first showed that small vessel cerebrovascular disease is associated with neurodegenerative changes in areas that overlap with some of the neurodegenerative changes that we see in Alzheimer's disease. And we think that these are partially tau mediated. She showed, again, statistically, a mediation that these areas that you see on this MRI scan, atrophy in those areas seems to mediate the association between white matter hyperintensity burden and cognition. And several years ago, too, Giuseppe Tosto showed that there was more rapid accumulation of tau pathology or tau markers in the CSF in people with higher baseline parietal lobe white matter hyperintensities. We did not see that baseline tau pathology or tau concentrations predicted accumulation of white matter hyperintensities. Rather, it seemed to be that the vascular lesions were pushing the tau concentrations up. In a recent paper from Crystal Lang, who is in my lab, Crystal first used plasma data to establish that there was a relationship between white matter hyperintensity volume on MRI scans with tau concentrations in the blood. And this seemed to be a stronger association in people who are symptomatic. And then we collaborated with Herman Moreno at Downstate and Frank Barone to create what we think is a sort of a white matter hyperintensity mouse model, where we conducted transient MCA occlusions and sham controls in mice. And first, we convinced ourselves that transient hypoperfusion indeed induces white matter hyperintensities. As we sort of index with myelin basic protein in mice, we see a reduction of white matter in occluded mice, and there's a dose response. The longer they're occluded, the more disruption we see. But more interesting to us was that in the occluded mice, there was an increase in phospho-tau relative to the sham controls. And these were in wild type mice, so they weren't beta amyloid mice and they weren't transgenic tau mice. Just in wild type mice, we could induce hyperphosphorylated tau pathology in the hippocampus on the ipsilateral side of the occlusion. And we thought that this was very interesting because we could essentially create Alzheimer's disease in the absence of amyloid. More recently, we examined the interaction among cerebral amyloid angiopathy with neuritic amyloid plaques, and we think that there's a very interesting story here. This was work done in collaboration with Jenny Rabin and Emma Nichols, and we had I think 3 key observations in this paper in this study. That first, this was with autopsy data. CAA and neuritic plaque densities actually share very little little variance with each other. That CAA seems to drive tau pathology, which in turn drives cognitive decline. In other words, tau mediates the effects of cerebral amyloid angiopathy on cognition. And that effect was particularly salient among those with high neuritic plaque burden. So there seems to be a statistical interaction, but maybe not a mechanistic interaction there. And neuritic plaques also drive tau pathology and cognitive decline, but that's also in the context of higher levels of CAA. So we think that neuritic plaques and cerebrovascular disease, at least here in the form of CAA, interact synergistically on tau pathology and its downstream cognitive decline. And this is what the data look like. These are across levels of CAA on the x-axis and the mean tau burden score on the y-axis, and you can see a very clear dose response. And this is the interaction when we interact CAA with neuritic plaque scores. We see that the impact or the association of CAA on tau pathology is much greater among those with higher neuritic plaque counts. So we've been plugging away at this model. It's probably wrong. We think that maybe CVD, cerebrovascular disease, might drive tau pathology, and I'd love to work a little bit more mechanistically and understand if inflammation and microglia response mediates this relationship in a feedback sort of way. We think that CVD and beta amyloid might interact, might not, we're not sure. And these interactions drive neurodegenerative changes, and that last square should say AD, not A beta. A number of outstanding questions. Do we think that these white matter changes are Wallerian? Is amyloid necessary? In other words, is there an amyloid independent pathway for the development of AD via vascular mechanisms? And a question that comes up a lot is, do we need vascular risk factors to drive cerebrovascular disease? And I think the answer is increasingly no, particularly from the genetic models, who are generally free or have very low vascular risk factors. So this has all led to a new grant that Liz mentioned yesterday. We're doing in collaboration. We're looking in Down syndrome to examine MRI markers of cerebrovascular disease. We're looking at some of these new blood-based biomarkers for vascular cognitive impairment, and then we're taking it to the lab and looking at some of the postmortem histopathological correlates of some of the observations that we're seeing in vivo. So in conclusion, I think that the current diagnostic models for AD are really insufficient for accounting the reality of the disease in the clinic. CVD appears to be an important component of the AD syndrome, at least by driving the phenotype and clinical incidence. CVD and amyloid pathology might interact in some critical way. CVD might contribute to dementia through tau hyperphosphorylation and neurodegeneration, possibly by inflammatory responses. And I think we can leverage neuroimaging to start to understand individual differences and sources of cognitive impairment and possibly treatment targets. So I think, evidence from late onset Alzheimer's disease, from autosomal dominant, from Alzheimer's disease and Down syndrome, all point to evidence that CVD is a core feature, and that we should really be looking at this a little bit more carefully. So I'll skip that just in the just for time's sake, and I'd like to acknowledge my current and past lab members. It's a really great group of people, and of course, my collaborators. Thanks so much for your attention. I'm happy to take any questions if we have time.
Thank you for all the clinical insights that you shared with us, Adam. Thank you very much. I think we probably only have time for one quick question. I guess I'll ask a question. So like you mentioned at the very end, there is little known genetic risk factors for vascular disease. So I just wonder if if there is any GWAS studies like done with the white matter hyperintensity like as the trait, so would that give us more power or not, and would that genetics results be different from any AD GWAS or not?
Yeah, that's a great question. So we just finished up an analysis with Richard Mayo and colleagues, and I forget the name of the gene, FMLN2. If I might be making that up, seems to be one of these genes that intersects between vascular disease and Alzheimer's disease, and obviously we have the cohorts to start doing in a hypothesis-driven way to see how these new GWAS identified SNPs associate with some of these imaging phenotypes.